Hi there, it's CEPA fellow Carrie Oderman. Welcome to the CEPA Podcast, where we bring together leaders and experts from both sides of the Atlantic to explore the biggest challenges shaping our world today. We hope you enjoy the episode.
Hello, I'm Alina Polyakova, President and CEO of CEPA, the Center for European Policy Analysis. Welcome back to the CEPA Podcast. This is such a special episode that I have the opportunity to host today because we're going to be talking about AI, bioengineering, biotech, and the revolution of AI semiconductors that is bringing to the entire world of science. And I am absolutely excited to be able to have that conversation with Dr. Kiana Aran, who is one of the world's leading creative thinkers, entrepreneurs, currently a professor of bioengineering at UC San Diego in California, but so many more other things, who is a technologist, an engineer, and someone who is thinking a lot about how the research that she's doing and the research that's happening in the broader field of bioengineering and biotech is transforming our societies. So, Kiana, thank you so much for being here. It's wonderful to have you on the podcast.
Thank you for having me.
So I want to start with a big question. And we're going to get into all of the super interesting work you're doing in real time in your lab out in UCSD. But the big question, I think, to my mind is, okay, like, what is biotech, first and foremost?
That's an interesting question. I think if you ask every technology developer who works at biotech, you'll hear a different answer. But for me, biotech, or biotechnology is a field where biology meets technology. So we create useful products and solutions by working with living, systems like our biology, our cells, our proteins, our DNA, or the whole organism. So we're creating solution for healthcare application. So to me, this is how I define biotech.
And AI, right? This is the big issue that often gets discussed in the national security world, where CEPA lives, where I spend most of my time. But when we talk about AI, it's data centers, it's energy, it's the race for the most sophisticated AI chips that companies like NVIDIA and others produce. But AI and the AI revolution is also permeating and having massive transformational effects in your field, in the field of bioengineering and biotech. What are the opportunities that you're seeing the integration and adaptation of AI produce in your field, in the biotech field?
I think AI has fundamentally changed how we do bioengineering, meaning how we engineer biology, or how do we connect with our biology, our cells, our DNA. So traditionally, when you think about biology, it's been very descriptive, very slow. We observe something, then we do experiment, and then we wait. But AI allows us to move forward much faster, sort of very predictive, and we can call it real-time biology. We can now model molecular interaction, predict protein structure, design or optimize the outcome of our pharmaceuticals, extract signal that are incredibly noisy from this biological system, and then put all of these data sets together and train algorithm that could do prediction. I think ability or capabilities that no human could have. This will allow us to sort of tap into decision making or information that we were never before able to, or if we were able to, it was very slow. So from that perspective, I think AI is really important. And in my lab, it's really not an add-on. We want to embed it into every measurement that we are getting. We want to combine high resolution technologies and sensors with AI to make sure that biology becomes something that we can access dynamically. So, and not just measuring the endpoint, because right now, the way we measure biology, for example, it's just like an endpoint of a process, and it's very slow. So I think AI will enable us to really, step forward and be much faster and more predictive. Of course, there are challenges with that, which I would be happy to talk about some of those challenges if you'd like. But I think it fundamentally changes how our healthcare system works. It fundamentally changes how we learn and understand biology. It fundamentally changes how we engineer, and it gives us the ability to engineer biology in a much faster pace than nature can do.
So I want to get into that a little bit because you alluded to, I think, two things. And I think for folks who don't have any direct technical experience or background, like I don't have any background in this field, but I find it so fascinating. And I think it really came together for me. One, when you just started talking about some of the challenges, but two, when I had a chance to visit your lab a few months ago, where you literally showed me some of the innovation that you're developing right now, that if one of these projects becomes commercial and scalable, would be like transformative for not just human ability, but for so many different elements of how we think about human evolution even, right? But let me start with the data, because data is also one of these big topics that we often talk about in the space of AI. But when you think about data as in biological data, like what is that really that you're talking about? And is that something that AI is having real integration with, right? What I mean by that is, like, I think we assume, oh, there's all these data centers, there's tons of data, and AI is getting better and better because it's getting trained in all this massive amounts of data. But how does that look right now in your field, right? Is there this mass data out there that AI is reading and producing some new innovative results around, or how is this really developing right now in bioengineering?
Yeah, no, that's a really good question. And the answer to you is no, or we don't have the data yet. And this is one of the major challenges that we're trying to deal with, because without sufficient amount of data, you cannot really trust, AI algorithm fully. So in context of biology, biological data are very, heterogeneous. They're context dependent. And we often poorly annotate them. What does that mean? Meaning that, when you're taking a biology of someone, you're giving a blood test, for example, and your biology is very different than someone else's biology who lives somewhere else in the world. You can't train your AI to just work on that data set alone. So it can be very different. So if you really want to have good, powerful AI algorithms, you really need to generate, a significant amount of data to make sure that you're capturing all of those, heterogeneity in data. Also, the way we interpret the data, it's a black box, right now to me, black box model do not translate easily to clinical or regulatory spaces. So how do we actually create some sort of a team of experts to basically sit down and interpret that data? And that requires years, and it requires data. The final thing I want to say in terms of challenges in my field, at least, it's integration of AI tools with experiment and wet lab. Most of the AI tools are developed, far from the wet labs, which creates a disconnect between what is being predicted and what is biologically meaningful. So right now, one of our main challenges is basically the format of the data, the amount of the data, and also validating the data. Is the data that, for example, AlphaFold is generating is absolutely correct in different cell and environmental contexts, for example, for engineering an enzyme? Is the predictive diagnostic that this AI model did can apply for someone who lives in Africa? So we can't have those answers because we don't have the data. The challenge is also in obtaining the data. For example, in our current clinical setting, if someone has, let's say, lung cancer, and we want to be able to use the data, the multimodal data from that person to develop an AI tool that can predict, lung cancer in someone else. The challenge is we have to find enough population that is heterogeneous. The second thing is right now, the way that we actually take that data, a person who have lung cancer who have to give a specimen from their lung, a part of that sample goes to a DNA lab, a part of it goes to an RNA lab, a part of it goes to a protein lab, a part of it is looked at by a pathologist that gives a qualitative answer. So the data format is very different. And then you're feeding a machine that is purely depending on digits and numbers, this data that does not have the same format. And it's very difficult to also obtain. So really, there is still a technological gap here that how do we put this data, create a format that is AI ready? And how do we generate enough data? And how do we actually put this system in our current healthcare workflow so we can obtain this data? So these are challenges that we're constantly trying to figure out solutions for. And, without that, I think we should expect, hallucinations from AI rather than like really true impact.
So sorry, go ahead.
No, I wanted to answer your second question regarding the superpowers you talked about.
Yes, I'm getting there. Can I follow up on that to kind of put a pin in it? Because you, on the one hand, it sounds like you're saying, look, AI is a transformational capability. But unlike in some other fields, where you can get these AI models training on a mass amount of data sets, we don't have that right now in the field of biology writ large, because it's just so much more difficult, right? It's like you're reading blood samples or x-rays, right? How do you transform that into AI readable material? But it sounds like when that moment happens, right, when suddenly you, let's say, have a data set, I don't know, of x-rays from every x-ray that's ever been taken around the world, right, in real time, the analytical capabilities seem, mind-boggling, right, what that could do in terms of diagnostics, in terms of being able to assess patterns and identify potential problems. And when you start getting into, beyond the very simple example of x-rays, right, looking at people's, genetics, right? I know that's already happening when it comes to bioengineering developments in genetic sequencing and things like that, right? I would love to hear a little bit more about like what future is out there, right? Once we get to a point where you have these mass readable biological data sets, like what are some of the things you think we'll be able to do? And I think one of those that is already happening, you showed me a little bit about this future, but that's not really sci-fi, it's actually here, is this whole idea of superpowers, right? This idea that we can harness the superpowers of other species, things that others can do that we cannot do, in a way that we used to not be able to do before. And that, again, gets us into another conversation about advanced chips and all of that. Can you connect the dots here a little bit? I know that was a lot, but like imagining a future in which these data challenges are less profound. And then where we are right now, already being able to really experiment with integrating these other species superpowers, like what are we talking about here really? And how's that going to work, right? Are we talking about some superhuman capabilities that we can even integrate into human biology? That starts to get, really interesting once we get into that. So over to you.
Yeah, thank you so much. Every time we talk about how do we connect the dots between, nature, AI, semiconductor, my heart starts, racing because there's so much excitement here. And my dream is to be alive for many years so I can actually see where this, synergize activities will land to. But when you think about using superpowers from other species, we have to also think about nature, right? Nature is another AI. It's just slow. It's a very slow AI that could generate, gets the data and then adapt based on the data. For example, us human, we have been adapted based on, what we have experienced, and our genetic codes have changed based on that. So it's really fascinating what nature has done. It's the most advanced technology out there. It's a bit slow, but it's very advanced. So other species have evolved amazing capabilities. And I'm going to put everything in a frame of example, so the listeners could basically connect the dots better. I always like to bring the example of olfactory receptors, because we all know that, for example, dogs or insects have extremely better smelling capability than us. The way smell works, you have this, we call them olfactory receptors. These are receptors on the membrane of the cells that respond to different odor and different smells. In insects and dogs, we have, very advanced olfactory receptors that could smell much better than us human can basically do. So we have learned about this biological system because we have developed, over the past 200 years, we have developed so many advanced technologies that gives us access to this biological data. From the invention of the microscope, the discovery of X-ray agent, that we can take X-ray and see what's inside of the cells in our body, from DNA sequencing technology and other advanced technologies, we have learned about our DNA, our proteins, the machineries inside the cells. And that have enabled us to now understand how the machineries work, what is the responsibility of each of these receptors, enzymes, machines in our cells in doing a certain activity. So now we're learning something from our biology and the machinery of this biological molecule, but we're also learning about plants, other animals. So then we can select the ones that are more superior, not overall, but in some aspect, like dogs and insects in smell. Identify the molecules that are responsible for that, and then harness them to basically create or maximize our capability in smelling. And where do we use that? You can imagine having a technology in your pocket that could smell better than you and gives you, information on your cell phone that, hey, I just smelled some sort of a chemical. We can do that, and that's not difficult. So the way I see this thing is that, how do we capture this biological information in something that human can read? And this is where semiconductor comes in. Semiconductor, or I like to call them transistors, because this is how we extract data from our biology. Semiconductor can extract, information that have some sort of an electronic element to it, right? Our whole body is an electronic system, because everything in our body has an electrical charge. Think about neurons. They communicate with each other electronically. But it's not only neurons. Every protein, DNA, RNA inside our body are electronically charged. So if we can allow our semiconductor to read this electronic information from our biology, then we can connect the two. Semiconductor can read when an olfactory receptor binds to a odorant or a smell, because that signal is an electrical signal. It's an electrostatic binding of two molecules together, which can be sensed by semiconductor and modern transistors. And our transistors are very fast. We can measure electrical signal at a much faster rate than biology. So we can really start tapping into reading this information that biological machineries are exchanging with each other in real time. Mm-hmm. So when an olfactory receptor binds to its smell, we get a signal in our sensor. Then we correlate the signal with what a smell we gave our system. And we train our algorithms that, okay, if you get this signal, this could be related to this smell. If you get this signal, you could be related to this smell. And the way you think about it, think about a parallelized, like an NVIDIA Blackwell chip that has billions of transistors. Imagine you attach a specific olfactory receptor that is specialized in sensing or smelling one odorant on the first transistor in the semiconductor. The other one, you add another olfactory receptor that is responding to a different smell. So now you start getting different signals from each of these transistors based on what they have smelled, right? Because they're very specific to what they're smelling. and train an algorithms that could tell you, okay, the combination of this odorant or these smells, it's because there is vanilla in there, or if this is this chemical in the air, or, you just found your partner, because actually moths, which are, insects, they kind of smell for their partner from kilometers away. But anyways, it's interesting that now with advancement in AI, semiconductor, and biology, you can combine these three together and start really tapping into capabilities that would be, unimaginable before. When you think about robots in future, I think this is how you can design your robot to smell. It could be really using or harnessing the superpower of other species, or if you could use other biological technologies to start, adding these capabilities to our, robots, or even, a system that we carry with us.
Yeah, so this is so fascinating because what you're describing is like this potential transformation in what humans could do, right? And not just humans. I mean, you brought in robots. I don't know how far I want to go into that direction, because it quickly becomes a scary thought to think about. I mean, a robot doesn't have to be a humanoid robot, right? But a robot that is actually much better at so many things than humans are, right? Not equal, but better, because it is much more capable of integrating chips, transistors, semiconductors into its system, and then it can, smell better. It can probably do other things better, like have, obviously higher strength, whatever we can think of as a human ability. But I think one of the things that you also have talked about when it comes to connecting the dots, and I saw so clearly when I visited your lab, is also how scale and the ability to scale and to synthesize biological material is like transforming these capabilities, right? So you talked about moths, and I didn't know this, but the fact that moths and their antennas have these very sensitive smell receptors, I had no idea. Also brings up some interesting questions about, okay, if moths can smell their close partners from miles away, like, okay, you have that capability in your phone. Okay, that's gets us into somewhat fun territory. But to go back to it, one of the things you said to me, is like, now we're in a position where if we identify this ability in a moth, for example, like we can take a sample, and it's not that we need to kill, a million moths to get this material. We can now synthesize this at massive scale. There are all these biofab capabilities. So again, in the world of, I think, traditional AI conversations, we talk about data centers and, chip production facilities, right? Everything we need to produce semiconductors and transistors. But there's this whole other scale up happening in the synthetic fabrication space, right? If I'm getting this right. And so this is also providing not just an ability to like identify a particular, enzyme or protein or a factor receptor, but then to mass produce it potentially, and have an application that can be commercial. And I know this is something you've thought about a lot, this question of not just doing research to progress research and science, but how do we actually get impact and usability and commercial applications. So I mentioned before that you're not just professor of bioengineering, but you're an inventor, an entrepreneur. You've had multiple companies that you started. You hold many patents of new technologies. So just to pivot a little bit, how do you think about this application side of it, right? Why is that something that you think really matters right now, that in the world of research, that there is more incentive or more focus on commercialization, right, of these capabilities?
Yeah, Alina, you added so many elements to this question that I want to make sure that I answered them correctly.
Answer whatever you want.
So I think the first thing that you mentioned I think it's really, really critical is my thought process was not like that if you met me 8 years ago, because as academic, we don't have that training. We only think about innovation. We only think about, answering the curious question in our mind, which is valuable, but also for me personally, until I got to the path of commercialization, a lot of this like thinking scale from early innovation days was not a part of my thinking. So that learning came from really being involved in industry and trying to commercialize my system. And I have really learned that if I want to make something commercializable and something to leave an impact, I have to think about the scale early on through my innovation process. So what you saw in my lab is that, for example, when we design sensor, we don't do one sensor. We always think about wafer scale. When we design an innovation, for example, even with the moth example you brought in, we work with a company that actually was produced those olfactory receptor for us at a scale, because otherwise, even if it's fascinating, if you can't scale it, then you can't kill moths to get their olfactory receptor. That's, you have to kill them, you have to, purify it, you have to extract and purify it. It's not doable. So it's really important that you think about the scale. And, but the good news is we are on that path. The biomanufacturing field has advanced so much. We had failure in US in terms of manufacturing, scale manufacturing in general. But I think a lot of people are pushing towards how do we actually not only engineer biology, because we can do that now with AI, but also scale it. And how do we make sure that we identify ways to purify the molecules that we want? Do we want to work with cells to make them produce, biology for us? Or do we want to actually take the component or those machineries outside of the cells and put them in bioreactor and may have them make very precise molecules for us? And I'm sure we'll get there. There are already companies that are working on cell-free biomanufacturing. So it's excitement. There's all excitement. You can design molecules now with AI. You can design them computationally with AI that could potentially actually behave better than the actual molecule in biology. Can we design an olfactory receptor that is more stable in high temperature as well, not only in low temperature? Because we live in different environment and there could be different temperatures, and you want that biology to be stable. You want the biology to basically respond in all of those environmental conditions. And we can do that with AI now. We can, computationally design those molecules in a way that they keep their function, but they're also more stable. So that's amazing that we could do that. And then we get to the manufacturing part. So once we design those computationally, we have to now manufacture them and test them. Mm-hmm. Testing now is a challenge, because when you test thousands of different, design of a molecule to see which one gives you the best answer and the best function compared to the biology you want to achieve from that specific molecule, the testing is slow right now, because you have to send them to different lab, different people to test them for that function and give you response back. So that's where the gap is right now, where if we have systems in place that after we design enzyme, after we manufacture them, and both of these areas are advancing significantly, can we have analyzer in the same environment that now it could test those molecules and provide a feedback loop to AI. So now you can have your machine running. If you can make everything autonomous, would be great. You can have your machine running, your bioreactor producing those machine design molecules. You have your analyzers testing those machine design molecules, then providing feedback to AI. You go home at night, you come back in the morning, and you have the molecule that would work best for your application, and then you scale that more. So it's again goes back to scaling, because I think scaling in biology doesn't just mean making more. It means that we learn faster, and that learning faster comes from this analyzer to assist AI in optimization instead of the trial and error, if that makes sense.
It does. It's such a complicated field, and I know it's difficult to connect all the dots, but it's what you just described, this entire supply chain almost, or ecosystem that's emerging because of innovation across so many different sectors. And the opportunities that holds are I think almost difficult to grasp for us, right? As we're talking about, people are just now starting to probably use like ChatGPT and other these like chatbots in their daily lives. But to really think beyond that, right? What is the future of biotech, right? And what does that look like? You started to allude to what I was going to ask you next. You talked a little bit about all the different pieces that have to come together, from the biomanufacturing to the basic research to innovation, right? And then the testing, and the system has to be efficient for you to be able to do that quickly. I wonder, in the policy space, in the geopolitical realm, these are issues that are kind of at the center of what we can call geostrategic competition, right? You're a scientist, you're a bioengineer. How do you see what's happening out there in the world? How do you think about, from a scientific engineering point of view, about the geopolitical landscape and where you fit into it, right? And I think the reason I'm asking that is because you alluded to this idea that the U.S. Is so good at innovation, right? And you're trained as a scientist to constantly focus on that. Like, where can we innovate? Where can we invent? Like, what is the entrepreneurial opportunity here, right? To do something different. And yet we have, I think you used the word failed, right? We failed to really maintain our ability to like produce, right? How does that affect what you do as a scientist, as an entrepreneur, an inventor, in your field?
That's a really good question. So I saw the direct impact of it during pandemic. So from one of our startups that is now acquired, but we were tasked to create chips that could help us in detection of COVID faster. We could have our chips basically detect COVID much faster, and the data would be digitalized. So it would be much faster to actually transmit this data to, clinics and doctors to make sure if someone is positive or negative. So we had great opportunities, but the government asked us to make, they asked us for a capability of making 2 million of these chips at minimum per week, which was way beyond what we could do. Because it was impossible for us to find foundries that could help us achieve that. And a lot of our parts were coming from, for our sensors and readers, were coming from different parts of the world. And during the pandemic, we had really problem in our supply chain, because we couldn't get access to what we needed. A lot of things were, held back for months, and it was really, really difficult. So it has a direct impact of how we actually translate all of this innovation that I talked about if we're still relying on other countries to provide us with tools and systems. And, historically, if you look, US have been amazing in innovation. From early days when transistors, in the semiconductor, when transistors were invented, when Gordon Moore, you hear about Moore law, that's coming from Gordon Moore, was trying to make the faster transistor, make them better. The Japanese countries were already using those earlier version of our transistor invented in the United States to make calculator or faster computers. This is when the investment, or the investors in Silicon Valley, this is when in the Bay Area, they basically woke up, and the Silicon Valley, this is how Silicon Valley started. Now we need to also produce something useful. But we failed to think about workflow processes, investing in how do we actually scale ourselves. We always relied on countries like China, because there was cheaper labor, and we provided them with our, IP and innovation for them to actually utilize it to scale for us. And that's a challenge, especially in current geopolitical, environment we are at. I think as a scientist and an engineer, I see the moment that we are in right now, both very fragile, and I think it's full of responsibilities. I think advanced chips are no longer just like a technological assets. They are geopolitical ones. For biotech, this raises an important question about supply chain resilience, scientific openness for us scientists versus security, and global equity in accessing innovation? How do we still continue to provide our innovation to a global, community? My view is that we need to do responsible innovation. We design a responsible innovation framework that protects national interest while preserving, somehow figure out a way to preserve scientific collaboration between people globally. And, we bioengineers sit at the intersection of health, security, technology, which means we must be more engaged with policy and not just retreat from them. And unfortunately, it's hard to convince scientists that being informed about geopolitical issues is important. As we are funded by our government to and it's our responsibility to also protect our nation.
I think that's an important point. It's probably the main reason I wanted us to have this conversation, right? Because we don't have the opportunity very often to hear directly from people like you who are at the forefront of doing the innovation, right? And you understand the challenges you're facing every day. You also see the opportunities and you know what the obstacles are. And you see how maybe different policy decisions are affecting your ability to continue to do your work. And I think supply chain resilience is obviously a big topic these days, as are some of the others you mentioned. But thank you for laying that out so clearly. So we're kind of nearing the end of our time, and I could talk to you forever about all the interesting research you're doing. I think we've only covered the very tip of the iceberg of this very, very big field and of your work over the years. You've done so much interesting work early on in your career that I've read about and learned about. So I think we'll need to have a follow-up conversation. But I wanted to ask you a little bit about you and how did you even get into this field? How did you get interested in this? I know that your path has not been, you could say, a conventional path. So tell us a little bit more about how you got interested, what your trajectories has been into this world of biotech and bioengineering there and now.
So that's a really good question. I think my path has been very not linear. So I'm originally Persian from Iran, and I did most of my early education in Iran. And back in Iran, if you were good in science, physics, and math, you would automatically become an electrical engineer because it was one of the highest engineering disciplines back in Iran. So when I moved from my undergrad study to US, I didn't even think about other majors. I directly went to electrical engineering, but I really never truly connected with electrical engineering because I wanted to see the impact of my work. So I was always drawn to problem solving, and I sat between disciplines of electrical engineering, biology, system engineering early on in my undergrad, and then pursued biomedical engineering for my graduate school and bioengineering for my postdoc. And it was not really until my postdoc where I started combining all of this field together and becoming a very interdisciplinary scientist. So I trained across different fields, and I learned how to take more risk, build platform that didn't quite exist. And of course, that meant a lot of failure, but also learning to speak a lot of different languages, academic, industrial, entrepreneurial. I think we all have our own languages. But what kept me really going was the belief that technology should be ultimately designed to serve human health. At least in my field, that's what my goal is. So that could have different, aspect of it in creating solution for some of the gaps that we talked about in terms of getting really real data from all of these AI design tools and trying to design sensor that could allow us to read biology more. So it's sort of very interdisciplinary. But I think I'm very agile. So if I find a problem, I tend to try to build solution around it. And that means that I collaborate extensively with people from different disciplines, and I truly enjoy it. So for me, I never have thought about anything that I have done in my life, in my career, as job, because this is something that's so close to my heart and I enjoy it so much. And I hope I can continue that enjoyment.
Well, I think as a part of this conversation, there's yet another language that you're adding to your multilingual approach to your work, which is, of course, language of national security and geopolitics. And I think it would be important for those of us in this field to learn the language of science a bit more as well, to really understand, one, I think how people like you have so much opportunity in this country in particular. And if you're intellectually curious and you pursue that, you have massive impact, and that's so unique to America, really. But also the current moment, the very fragile moment for all the reasons you mentioned, where there is not just intensified competition for innovation, but intensified competition for talent, right? And the opportunity that we have in this country to maintain our ability to lead on innovation also depends on our ability to be able to still have an open environment for talent to thrive, to have collaboration and research, which is, very much part of what is at the heart of CEPA as well, right? To be an institution that provides those kinds of opportunities to experts, to people who are at the edge of thinking about their issues. Kiana, thank you so much for this conversation.
Absolutely. Thank you so much for having me. I just wanted to add something to what you said, because it's actually very close to my heart. I think one of the things that draws so many people to the United States is the land of opportunity. And it's the true thing, because if you are motivated, it's an environment that attracts people who have good and right motivational factor. So if you could channel that somehow to what is different, motivational factor for different people, and how do we create a society where, keep people aligned with motivational factors in a way that really advances our national security or innovation, or, thinking that way, I think it would be very, very powerful. Because if you look at the majority of us who come to the United States, we find this where we can really give back to the country that have accepted us as because I'm an immigrant, have accepted me as an immigrant and provided me with tools where I can then, give back to US, and I'm very grateful for that. And I think, thinking a bit more broad about how do we think about, attracting talent, and being aligned with their motivational factors, so then we can fully benefit from the talent. It would be very important.
Absolutely, I couldn't agree with you more. It's the foundation of this country in so many ways. Kiana, thank you again for your time. I know we could have talked for a long time. There's so many other interesting, innovative research projects that I know you're working on right now that I wish we could have gotten into in this conversation, that just completely blew my mind when you told me about some of them when I saw you in San Diego, your lab. So we'll have to have you back on. But thanks again. This was absolutely fascinating conversation.
Thank you very much for having me, Alina.
If you enjoyed listening, be sure to subscribe so you never miss an episode. You can find us on Apple Podcast, Spotify, or wherever else you get your podcasts. And to dive deeper into the issues shaping transatlantic policy, visit CEPA.org, where you'll find more expert analysis and research. And don't forget, you can find us all across social media for the latest updates, thought leadership, and exclusive content. Thanks for listening.